one. So today I wanted to continue with the theme of information theory, but raise the question of is it useful in the brain, and if we believe that that's the theory of communication and of extracting signals from the environment and representing them in an efficient manner, then it must be true that these principles should also guide the operation of real small neural circuits. And so today I will describe a project that was done in collaboration with Chelasani's laboratory at the Salk, and by our student Adam Calhoun, who recently graduated. So this piece is part of his thesis. And I tried to give a broad title of Information-Theoretic Principles in Learning and Decision Making. And it all began for me with this observation of this interesting behavior of C. elegans. So C. elegans is a small worm about 1 millimeter in size. And what is interesting about it is that it only has 300 neurons. And if you think about how many sensory neurons, well, it just maybe under 10. So it really pushes the limits of what this animal can extract, how they can operate on the environment, but nevertheless, they still need to figure out where the food is. They need to build potentially complicated models of how this food is distributed, how to detect it, how to avoid predators. So what I will describe today is that their behavior actually conforms with information maximization principle. So they're driven by maximizing information. But at the same time, while these calculations are very complicated, I will show how they can be approximated in there are ad hoc ways of approximating the optimal solutions such that the animal can actually implement it in a small neural circuit. So this is an example of the behavior that is easily quantifiable. And so what is shown here are traces of a worm as they crawl around on a plate of agar. And on this particular plate, there are bacteria, and C. elegans eat bacteria as they move around. So you can see that when the animal is on food, they make lots of turns. An interesting thing happens when the animal is picked up and moved to a different plate that has no bacteria and no food. So initially, they go to a place where they think the food was, and they make lots of turns. And then at some point, after half an hour or so, they make essentially straight motion away from the area and explore a new domain. So an interesting feature about this is that that the time that they search for food is stereotypic, but it's also modulated. If the food was tastier, they will search for longer. And also, so the question is, it's very reminiscent of how we would behave. If conditions are good, that's okay. If the conditions are bad, I will try to change them for some limited period of time, but if it is not succeeding, then we should switch to some other strategy. So I've used this as an element of decision making, as a decision making motif that potentially could be used in the nervous system over and over. So this is a description of the same experiment of now you can see the whole plate where this is a drop of bacteria, so the worm crawls around. This is on a plate without bacteria, what we call the initial phase, the local search, and then the comparison between the area that they explore during the local search and the area they explore during the global search. So that's an example of a behavior that is very important to the animal and yet quantifiable, and also challenging because they need to find food over the area that is many thousand times larger than their body size. So one could argue how can we begin addressing this problem? And at the time we were starting to work on this project, there was this documentary by Stephen Hawking, and he said that, what are we doing beaming electrosongs into space, attracting attention of aliens? If they're going to come, it's not going to work out well for us. And I was discussing this movie with my husband over dinner, and he said, well, why do we ascribe the properties of humans to aliens? They could be completely different. And at the time, my point was that they're not going to be different because there are likely fundamental rules based on information theory of how any creature should behave, regardless of what they're made from. And we can't make experiments on aliens, but we could make experiments on C. elegans. And so that was another part of this interest of trying to understand what are the rules that guide behavior. And it turns out that there were previously two separate theories that are actually quite related to each other. And one theory was, one strategy was termed infotaxes. That's as opposed to chemotaxis that some of you may have heard about. So chemotaxis is a classical model of navigation for single-cell organisms. Also within the nervous system, neurons can find their targets by chemotactic cues, navigating towards the molecules, finding the gradients of the molecules that they are attracted to. But in addition, instead of estimating the gradient and moving in the direction of the largest gradient, one could design a different algorithm called infotaxis, where at each step the searcher chooses their next move according to the degree to which that reduces uncertainty about the target location. And uncertainty we are going to measure as entropy of the probability distribution. So the change in uncertainty from the anticipated move is how much information I expect to gain if I make a move to a given direction. So in this case, that's the setup of the algorithm. And the idea is that, for example, I can be at one location and I project that I have a mental map of what is the probability distributions of food sources throughout the environment. Based on this probability distribution, if I make a step to a given location, what's the probability that I will detect an odorant? So I need to have the diffusion model of how odorants move from targets to that location. So once I estimate the probability distribution of detecting an odor at a given location, I would then say if I actually detect an odorant, how much information I would gain, and if I don't detect an odorant, how much information I would gain, because that's also informative. If I think I'm close to the source and I'm not detecting any odorants, then that also carries information. So one could begin to solve these search strategies, and in the case, originally it was proposed in the case of navigation of odorant. There is a real source and odorants diffuse, and here the searcher begins here, and all the dots are where the odorants are detected. So this algorithm could reproduce interesting features of animal behavior, such as crosswind zigzagging, and where the animal is trying to find plumes. And that's more shown here. In the case of a larger wind speed, the searcher begins here. They find the food source at the end of the trajectory, but when they do not detect odorants for a long time, they start making the zigzag motion perpendicular to it. So that's one example where information theory is successful, and it could explain how moths find their mates over large distances. So let's consider a very different example of visual eye movements. So that's towards the general theory of decision making. And in this case, the setup is very different, because instead of crawling continuously through space, I can look at discrete and different locations in the environment and collect information from this. So the physical setup is very different, yet another group proposed essentially the same algorithm, but for deciding where we are going to look next. So in their experiments, the subjects were asked to find a target, and I think they find at after forced eye movement. But the initial is that when the subject looks in a given place marked by a white spot, the probability that the target is nearby is reduced. It's similar to the diffusion kernel, but now it's because of the visual resolution of the visual system. If I look in a certain location and I don't detect the target, it means that it's not nearby. So, and then once we look in a different location that sort of zeroes the probability here, but maybe there are two peaks. So the subject explores one peak and then finally finds the target at the other. So in the case of visual search, this information maximization algorithm could also account for interesting features of human eye movement behavior, because sometimes humans make what are called elimination saccades, meaning eye movements in between two possible targets. And a key feature of the infotaxes compared to the chemotaxis model is that they are not mutually exclusive, meaning that close to the source, information maximization becomes same thing as a gradient, chemical gradient. But in cases where the signal to noise is not reliable, then it's better to maximize information directly as opposed to secondary quantities. So we wanted to test the generality of this framework in our system, so which is warm behavior. And the setup is actually quite different, because in our case, there are no odorant cues. So the animal thinks everything is based on the prior belief of the animal. So that's the only driving force for this algorithm. There are no odorant detection cues. The animal expects to find odorants, but actually it never finds it. So this was the result of simulations. First, in simulations, we assumed that the animal has certain prior, shown here as color, as a Gaussian distribution with brighter colors representing higher likelihood to find food there. The searcher starts here and initially moves towards the peak of these distributions, and then the trajectory follows roughly a spiral. One can show that in the continuous limit, it's actually an Archimedean spiral. And then, at least at some point, however, at some point, the spiraling motion stops and the searcher moves directly towards the boundary. So this is very reminiscent of what is observed in the experiment. We would associate that the spiraling part of the trajectory with the local search or area-restricted search, and the straight motion towards the boundary as the transition to the global search. And so that matches some aspects of the behavior. And then we decided to test this quantitatively also, so such that here is the probability distribution of C. elegans position at the end of the local search. The dashed line is for experiments, and red line is for the model. And they match well, but we had to adjust some parameters. For example, the diffusion constant for the odorant, and most importantly, the prior, the width of the initial prior distribution. And but once we adjust these parameters, we could then predict another part is the time distribution. How long did the local search last? So that also agrees. There's agreement between theory and experiment in terms of the how long the local search we expect to last. So these are data combined. This is a cumulative distribution combining data across multiple genetically identical animals. And so that's encouraging. However, there are some issues that we needed to follow up on. And one of them is that in the model, the transition between a local search and a global search is abrupt, whereas in experiments, if we observe the number of reorientations per minute, which is roughly, the more they make the turns during the local search to the global search is more smeared. So the question to address was whether this discrepancy is due to the fact that we averaged across many different worms, many different experiments. So one could answer that question by applying hidden Markov models that to the animal trajectory and examining what is the time with which the state changes from local state to the global state, global search state, and the switching time is actually quite short, as inferred from the experimental motion, experimental data. So finally, one could compare predictions between the duration of this, the transition of the duration, how fast the animal transitions from local search to the global search, with the prediction of what I would call a more classically a single time constant adapting process, or chemotaxis. And I will say in a moment why this is related to chemotaxis. And we will find that the transition duration is much larger than what is observed experimentally. So the, at this, often the question arises is, could a conventional chemotaxis model explain the animal behavior? Because initially the odorant concentration is high, and then the odorant concentration is low. That would induce the number of turns initially, and then as the animal adapts to the new zero concentration, that number of turns will decrease. But the key feature is that if I assume a single time constant, I can either explain that the transition is short, or I can explain that the duration of the local search is long. So in fact, we have a more gradual process that is more step-like and then goes to the plateau, and it's more difficult to explain with an adapting process. So that's shown here. Initially, based on the initial, if we estimate the rate of decay based on the initial decay in the number of turns, that would predict a much larger turning rate than is observed experimentally at the end of the search. On the other hand, if you want to examine the final turning rate in the baseline, we will not going to capture how fast the transition from the local and global search. So that's one evidence that the search strategy they implement is more complicated than a chemotaxis. Yes?
So can you say again what you mean by single adaptive process?
So the conventional model is that suppose a neuron, an animal has a sensory neuron, and which they do, and the sensory neuron will be activated by the removal of odorants. So that sensory neuron will produce a signal, and then if the number of turns is controlled by the activation of that neuron, that number of turns will decay. And usually the canonical, the simplest model is that it decays according to an exponential with a single time constant. And so that's what a behavior that one could achieve using kind of based on one sensory neuron. But the trick is that the sensory neuron gets activated, but it stays active only for a very short, relatively short period of time of under 1 minute. And then whereas the local search behavior lasts 15 minutes. So the underlying question is, how does one convert a short-term signal to a more long-term memory, but then which itself erodes. So, and another prediction that was qualitatively different between infotaxes and chemotaxis is the dependence on the concentration of the food patch. So the key model in the infotaxis prediction is that the animal is driven by the probability distribution of how food is likely to be distributed. It's not driven by the absolute amount. In the chemotaxis model, in contrast, if I accommodate animals to a patch of food with high concentration, when I remove the animals from that patch, they should have a larger number of, they should make larger number of turns. So chemotaxis predicts that if we use either normal bacterial concentration or diluted, there should be a drop in the number of turns. Yet in the infotaxis model, experimentally they're identical, and that's one of the key predictions from the infotaxis model. So that's, to me, that's one of the examples of a rudimentary invariance that is developing because the circuit is, as I will argue, a little more complex than just one sensory neuron, although I will say that this is what you can get with actually 3 or 4 neurons. So moving forward, and the question arises is that what I have described is an algorithm that numerically is difficult to implement. I have to memorize the position, keep in the memory the probability distribution of food. I need to update it as the searcher moves along, and in principle have infinitely long memory. So how can this possibly be implemented in a small neural circuit? So one answer to this, one clue we obtained when we looked at what happens to the probability distribution that food is not located within the immediate area, but is actually located elsewhere. So initially, that log probability that the food is located is somewhere else is very small. So it's and then, but as the searcher moves around and explores the central peak of the distribution, this probability is updated in a Bayesian manner, and it increases. And it turns out that the transition from the local search in the model to the global search occurs when that probability that the food is located elsewhere reaches 1. So that provides bypass to the maximally informative calculation, because instead of doing complicated calculations, all I need to do is to estimate the rate with which this probability distribution will change in time. So a sensory neuron could trigger the start of the search. Another neuron could keep track of time as a function of and then when that neuron reaches voltage reaches the threshold, then the transition from the local search to the global search might begin. So that's the working model, and this model is actually flexible. So these are predictions, parameters of the infotaxis model, but as I mentioned, they depend on whether the prior is more narrow or more wide. By prior, I mean that if the worms were raised on a patch of food that had small extent versus large extent, and in this case, if they were raised on a wider distribution, then they should be, the prediction is that they should be searching for longer. And actually is observed experimentally. But interestingly, we can approximate the full information calculation with just a drift diffusion model. And by adjusting the slope of this drift diffusion model, the animals could mimic or approximate the information-theoretic solution. So, and how should I finish? Okay. So, in fact, there is a uniform scaling by the probability distribution, by the widths of the probability distribution, such that the wider this, the initial prior, the longer it takes for the animals to search. But if we rescale this time by the sigma of the initial distributions, all of these curves overlap, and therefore the animal just need to learn the slope, perhaps through experience, in order to implement such a strategy. And I think I will skip this in the interest of time, and because I want to cover one more piece, outline maybe potentially future directions. So the working model that these data suggest is that instead of driving the behavior based on one sensory neuron, the circuit can be made a little bit more complicated with 3 neurons. And we have a sensory neuron to trigger the start of the local search. The evidence accumulation will be measured in one of the interneurons. And then when that neuron triggers the search, reaches critical voltage, that would trigger the transition to the global search. And there are other neurons in the circuit through neuromodulators that can modulate the rate within which the evidence accumulates. So that accumulation of evidence could be adjusted depending on how hungry the animal is, how tasty the bacteria are, and through experience, if the patch is wide, how long does it make sense for me to look for that, continue to look in that area. So at this point, we wanted to look at whether we could examine variations in animal behavior. So as I mentioned, the experiments are done on genetically identical animals, but nevertheless, there is a fairly substantial degree of variability in the number of turns. Some do the local search complete shorter, some longer. On average, we could explain the probability distribution. But the question is, can one account for animal behavior on a moment-by-moment basis? And perhaps the reason is that they had different prior sensory experiences. So then it turns out that more detailed experimental measurements show that the patch of bacteria is actually not a Gaussian, like we initially assumed, but it's more of a ring with a nearly constant concentration and a larger concentration at the edges. And then one could image the worm motion while they're acclimated to a patch of food, so that's the worm trajectories, and measure how often they were on the edge and therefore experienced large change in the bacterial concentration. So taking that prior time history, one could then build a model of how how the animal should behave based on a particular sequence of sensory experience. And that turns out that once you take that into account, that explains a lot of the data. So let me show you the steps of the analysis. So as inputs, we will take times on the edges of the bacteria, times when the animal was on the edge of the bacterial lawn. And the output will be the number of turns when the animal is removed off food. So this is prior time, and then it's prediction into the future. And then we will find what is the optimal kernel or optimal filter that relates the memory of past sensory experience to future number of turns. And that kernel actually has an interesting, from the biology standpoint, interesting features. It is 0 for the past 5 minutes, meaning it takes at least 5 minutes for the sensory experience to affect future behavior. It's also 0 for very long times. So according to this statistical analysis, which was done by kind of a linear, nonlinear model similar to what you heard about in the previous talk, the maximum impact that the sensory experience has is about 30 minutes prior, 20 to about 15 to 20 minutes prior to the animal being removed from food. So one can test this directly by letting the animals acclimate to bacteria with large distribution, and then either transferring them to large distributions or first to small distribution and then to the large distribution. And depending on how long time they spend on the small patch, we can see whether they have adjusted, memorized this spatial extent, or their behavior is primarily driven by the large extent of the patch. So if they were on the small patch for 5 minutes, they behave as if they were never there. But as long as they are for 15 or 30 minutes, their behavior matches the small patch experiment. So that's an independent verification of the filters that were derived using minimal models. So we fit a using logistical regression. Relating times on the edge to the number of turns. And with this model, we can now compare how many turns the animal made. So there is a substantial degree of variability between 10 and 80, and compare that with model predictions, which are highly correlated. So I think that brings me to the end of my talk. So what I wanted to argue is that Bayesian search strategy, or information maximization, can account for the transition in animals' behavior. And I would argue that it made several predictions such that actually will be tested. So one of the predictions that it should depend on the worm size. So bigger worms should make their decision faster. And it increases with width of the prior distribution. And I hope, and then the variability in sensory experience can be explains the variability in future behavior. So sometimes we think that the person might behave suboptimally or in a strange way, but actually is perfectly optimal given their past sensory experience. So with this, I would like to acknowledge my group for their many stimulating discussions, and again, the Adam Calhoun and Srikant and our funding. Thank you for your attention.